monitoring pollinators and their stressors. This panel that we have now, it tends to make an overview of some of the European research projects gathering data of the status of EU pollinators or the stressing factors that can affect them, such as the environmental pollutants, pathogens, the landscape, weather, etc. And with this introduction, I would like to give the floor to Mr. Martin Heusig for some introductory remarks. Thank you.
Thank you very much, Noah, and I'm really pleased to be able to open this session and host it. And I'm very pleased that you are facilitating the entire session, because the collaboration with BeLive has been really, really important on that, during the whole Pollinators Week, actually. Now, as a European Parliament, we've been quite vocal in saying that we need to not only protect bees and pollinators, or all of the pollinators, but we actually need to know more. We need to have a better data. And this is my first legislative term as a member of the European Parliament. But even in this short 2 years, during which we had to also face pandemic, there was a number of initiatives, resolutions from the European Parliament on the topic of the pollinators. And I'm also happy that we managed to get through, and I'm kind of a bit proud also that I'm behind some of the what we call pilot projects and preparatory actions on the monitoring of pollinators in Europe. But let's be honest, Insignia, which is from the previous mandate, from Pavel Potts, as well as Spring, which is the result of the pilot project that I initiated, are not the only things. And that's really, really good to hear. It's amazing that we also will have here a presentation that will discuss projects from European Environmental Agency, the POSHBI project, Be Good, EMBAL, but there are others. And I think this is something where we have to be clear that it should not be only about gathering more data. We need to do it smartly, and we need to collaborate better. Just on Monday, I heard from the State Secretary for Environment, so the Vice Minister of Environment from Slovakia, saying, yeah, they are essentially starting their own monitoring of pollinators, and during a workshop, and they will try to somehow kind of connect it to the European one later, which I think it's a pity that they're not part of the Spring and from the start. And it's clear signal that we have to do more in Europe to connect all these projects, to exchange practices, and to have kind of a better wealth of data. The similar project in Ireland. What I learned on Monday in the kind of the Be Political session, and I recommend you to watch it again, though it was tough to get the member states speaking, I have to say, that they are doing quite an effort in monitoring pollinators. And clearly we're not doing enough to kind of connect all the dots. It doesn't mean that we should do less, actually. I think we should be better in identifying what are the missing links, because then when you talk to EFSA, when you hear about the challenges we have in terms of the non-bee pollinators and how we assess them, and kind of how we see and monitor the impacts, and how we kind of assess the impacts of, for example, pesticides on them, we don't have enough data. So this is something where I'm very grateful. I'm really looking forward to hear more from all of you about the different projects. But I would like to encourage you also to say, okay, how we can work better together, because this is one of the incredible strengths of the European Union, that we actually can do things together and can deliver way more for citizens, planet, and ultimately, therefore, also for our own prosperity and future. Now, I will finish here and hand over back to Noah for the session. I'm really looking forward to hear more about the data, your plans, but also your obstacles. Please share with us where you see the challenges, and I would be particularly happy to hear where Parliament but also the other institutions and the member state governments could be better. What can be changed to make your work easier, to make your work more efficient, to help you to collaborate and to help you to deliver more protection and actually sufficient protection for all the buzzing creature out there, which at the end of the day keep also us alive. Thank you very much.
Thank you very much, Martin, for these inspiring words about also the need and the invitation for collaboration and of sharing, and for the openness of the institutions to answer to the needs of citizens and researchers in their daily life. Okay, with this, I would like just to remind some housekeeping rules. So I see that you are all well experienced in participating to this kind of event. So everybody's with the camera off and muted, so that's very good. I ask the attendees to keep like this and to please write their questions and suggestions in the chat. And yeah, and then we will take them for the relevant speakers. And with this, I would like to introduce Dr. Biala, Katerina Biala, from the European Environmental Agency. She's a the biodiversity indicators and assessment expert, and she's going to present us the activities that the agency is doing to build up knowledge base about pollinators and to reverse their declines. Dr. Biala, the floor is yours.
Thank you. Good morning, Mr. Chair. Good morning, everybody, and thank you very much for the invitation today. So, European Environment Agency is one of the EU agencies, and our objective is to provide information on the environment in support of policy, as well as to the general public. With regard to knowledge about pollinators, I would like to start with sharing. I'm sorry, doesn't. I can't seem to be moving my slides.
Okay. No,
sorry about that.
No stress. These are the issues of the direct.
Yes.
Otherwise, I'm going to try to share them from here. Okay. So for you, it's not moving, eh?
No.
Okay, so then I'm going to share my screen. And I'm going to make it, you will tell me when I have to make it, when I have to change it. Can you see the presentation now?
Yes, thank you. So please, next slide. One of those policies is, of course, the EU biodiversity strategy with the clear aims of protecting and restoring nature. And with regard to pollinators, one of the things we have been doing is sharing knowledge. Next slide, please.
Oh.
Thank you. So since 2010, we have been publishing on our website the grassland butterfly indicator. It has been developed by NGOs, scientists, and literally thousands of volunteers. You will have a chance to hear more about butterfly monitoring in a later session today, so I will not go so much into detail, but I will only highlight the strongly decreasing trend within this period, 91 and 2018. It was a 25% decrease of the index. But it's also a perfect opportunity to express our deep appreciation to all those who have developed the indicator and have been contributing to it over the years. and also thanks to whom we, as the EA, were also able to share it more widely among different audiences. Next slide, please. Now moving to what have we been doing recently to build more knowledge about pollinators and using Habitats Directive, which is one of our core areas of activities. So the Habitats Directive protects both habitats and species. And every 6 years, member states report on the conservation status and trends in targeted habitats and species. This is this Article 17 reporting. It's both national institutions as well as volunteers, citizen scientists. So it's a more inclusive process. And we, the EEA, and our topic center on biological diversity have been providing both technical and scientific support to the Commission, but in particular also to the member states throughout all stages of the reporting process. And this wealth of information is then in turn published in our EEA report, State of Nature in the EU. So here you can see the last report, which we published in 2020. And for this report, next slide, please. We have been considering how we can deepen, refine it, and how we could use this really unique information built over the years through the Habitats Directive to add, enhance our knowledge about pollinators. And a logical question, and question that would need to be answered, was which habitats are important for pollinators, both as sources of food and as nesting sites. Next slide, please. Thank you. So our partners from the Topic Center on Biodiversity took a deep dive into Annex 1 of this Habitats Directive and compiled a list of well over 6,500 vascular plants that belong to these habitats, and discovered that quite a lot of them, nearly 70%, can be classified according to their importance for pollinators. And then that more than half of them are actually important for pollinators. So this was a really refined analysis published in a report, and just the most important conclusions that the Topic Center and ourselves took from this work so far. So we actually know much more about which plants are suitable as food source, but much less about the nesting site abilities. Taking into account all the outcomes was a conclusion that plant species richness is the best proxy variable to assess the habitat type importance for pollinators. And if we wanted to think about the top 3 habitats that are really of importance to pollinators, they would be grasslands, sclerophyllous scrubs. So for example, these are garrigue or matorral in the Mediterranean region, and also temperate heath. They would be these top 3. The next slide, please. So the next step, okay, we know which habitats are important for pollinators. So how can we combine it with Article 17 reporting results on the conservation status? Here you can see the outcomes of the 2020 report for all these 233 habitats, not only those important for pollinators. And as you can see, the situation is that only 15% of habitat assessments have a good conservation status, and as much as 81% have poor or bad conservation status. So how do the habitats important for pollinators fare? What is their conservation status? Next slide, please. Well, actually, we found out that they have an even worse conservation status and trends than other habitats. Part of this information is in the State of Nature report, but we also built an online dashboard on our page Explore Nature Reporting Data. And now I would like to present a few screenshots which would give you some more information. Next slide, please, Noah. Thank you. So here you can see the conservation status. Again, a lot of red, a lot of orange. Definitely not as much green as we would like to see. The situation really needs significant improvement. When we talk about the trends, which the countries also reported on, next slide, Noah, please. The situation is similar. A lot of this purple color, which indicates deteriorating trends, and definitely not enough assessments showing improving trends. So that's the conservation status and trends, but yes, thank you, the next slide. But countries also reported on the pressures for habitats and species, and thanks to this, we could also analyze more deeply threats for pollinators and pressures. And as you can see here, in particularly visible for grasslands, but also for practically all other types of habitats, agriculture exerts the most pressure, and it's both abandonment, like cessation of grazing and mowing, or intensive grazing or overgrazing by livestock, and in general intensive agriculture, which, as we know, also entails use of pesticides, which was clearly one of the pressures and threats to pollinators, both domesticated and wild. The next slide, please. So we also made a first attempt to start mapping those habitats. Here you can see the rough mapping of grasslands distribution, those that are important of pollinators, because of course to assess the status, but also to think about the solutions, we also know where these habitats are. Next slide, please, Noah. So, outcomes and conclusions. First of all, our aim was to fill some knowledge gaps on pollinators. So we started to build a source that could be used by other ongoing and future projects, assessments and policy evaluation, which could be used as information about habitats, but also in the wider frame, next slide please, as indicating solutions. We know that many actions are needed to reverse pollinator decline. It's of course about the numbers, but of course the numbers are also heavily influenced about the quality and status of the habitats and the environment they are living in. So as I said, it's not a purely academic exercise. It's a very pragmatic task that we tried to develop so as we could help a lot of different users that could take it as part of their own assessments, as part of their own projects. As I was saying earlier, we have been working on those lists, and we are continuing to refining them this year because we heard that this could also be needed. And so we also wanted to put it in this wider frame that protection and restoration of the habitats for pollinators will also benefit wider biodiversity as well as our societies and economies. So thank you very much. And with this, I would like to thank for your attention.
Thank you very much, Katarina. So at this time, I'm going to check in the chat if there are questions. No, okay. So I would like actually to ask, sorry, I would like to ask you a question. All these data that you have being collected to build up all these maps, etc. is it also available? So it can be usable, let's say, by other groups.
Yes, definitely, very much. So we publish all the outcomes of the analysis on the EA website. So this can be accessed by general public. This doesn't require any special registration. And of course, this work is also a part of what the Commission, in particular DG Environment, is also sharing on their website. So it's this combination of efforts and moving the reporting into the assessment part, where we can not only show the results, but try to also indicate on some solutions and actions that would be needed.
And if I may, so now, well, we are going to have a big amount of data, of initiatives that are going to be produced, let's say. These, like, how do you envisage, for instance, these connections or these integrations once the data is available?
Thank you. That's a very good point. I think we are already building on our past activities, because it sort of comes organically. You discover initiatives, you try to link to them, and also linking to the initiatives that Mr. Hojsik mentioned in his introductory address. So we are already following the Spring project. We are already following other Horizon 2020 projects, exactly to provide this added value. So it's the collaboration and building on each other outcomes and results, with the purpose of not duplicating the efforts, but exactly providing the added value and building this collaboration. You are right. Of course, there are many of those initiatives. It's impossible to follow all of them, but it's absolutely necessary to make this effort to link and to think of pollinators as parts of wider biodiversity, of wider environment, and use them as the group that could also involve citizens and businesses, for example.
Sorry, I'm just going to take a question from the audience. Sorry, Rainer, because afterwards you will be able also to discuss. So by restoration, is Piotr Metriki, by restoration of their habitats, do you intend also intervention on the structure of agriculture or only natural habitats?
It's not only natural habitats. Of course, pollinators are intrinsically linked to agricultural habitats. Even if we think about Habitats Directive, of course, Article 17 reporting is interesting because it's not only restricted to protected areas, but it goes much wider into the European territory. But on the other hand, even if we think about Natura 2000 sites, which are also a key part of the Habitats Directive, more or less 50% of Natura 2000 sites are linked to agriculture. So it's definitely, and they are used. They are not pristine areas. They are areas that are being used. So I think addressing the restoration of habitats for pollinators will undoubtedly have to involve agriculture as well.
Okay. Thank you. So we need to move on to the next presentation. Katerina, maybe you have access to the question and answer, so maybe you can answer in writing. Thank you very much. And then all these question and answers, they can be made available when we do the report of the session. So with this, I would like to give the floor to Dr. Dalel Askri, who is a postdoctoral research engineer at the Biopark d'Archamps in France, and is going to present us the monitoring of stressors performed within the project POSHV. Thank you, Dalel. The floor is yours.
Thank you. Good morning. Thank you, Mr. Chair. Thank you, Noah. To start, do you see my screen?
No.
Oh, just a second. maybe I had a...
Now we can see it.
Okay. Okay. To start, POSH-B project is a Horizon 2020 consortium project led by Professor Mark Brown at the Royal Holloway University of London. POSH-B is a large project with 42 partners and many aims, but here today we focus on our work on monitoring bee stressors. Among work packages, the 13th work package of POSH-B, we have the work package 9, which is related to omics, and we are developing an innovative tool for bee health monitoring, which is a proteomic-based bee health card. Within this work package 9, we are using mass spectrometry to analyze bee blood, which is hemolymph, collected from different species, mainly honeybees, bumblebees, and solitary bees, to monitor bee health following exposures to different stressors, such as abiotic and biotic stressors, and from different experiments within POSH-B, from laboratory, semi-field, and field experiments. The first technique we use is the multi-bee typing, and it aims to generate mass fingerprints from individual bees and then create models to classify the stressors. The second technique is the bottom-up proteomic approach that aims to identify and characterize stress markers using the label-free quantification, LFQ, and also gene ontology and biological pathways analysis with bioinformatics tools to decipher altered pathways linked to stressors. The third technique we use is the mass spectrometry imaging, which is a technique that allows us to spatial mapping of specific molecular markers in bee tissues. Now, how to generate the health card? As I said before, we have the multi-bee typing, which is the basic technique for the omics part of this project. After a reception and analysis of hemolymph samples from laboratory experiments within work package 3 and 6, we generated the mass fingerprints, and these mass fingerprints are used to elaborate stressor models, such as for agrochemicals, nutriments, and pathogens. And later, from a given bee, we will confront hemolymph mass fingerprints with the library that we generated based on our experiments in POSH-B to evaluate each stressor impact and the human response level of the bee. As you can see here in this scheme, the beekeeper will collect the bees or the products from the hive, then send it to an analytical laboratory to have the mass fingerprints analysis and the model confrontation, and then generate the bee health report, which will be used as a multi-scale monitoring tool for sanitary policy and beekeeper best practice. Now, let's move to the second tool developed by POSH-B partner ANSES and University of Bordeaux in France here within the work package 2, the passive air sampling inside beehives. Classically, in order to sample atmospheric pesticides, active pumping systems are used, but these systems are expensive and require training operator that needs to be sure about the installation and the maintenance to perform sampling. Moreover, these techniques would disrupt bee colonies. So the tools we are developing need to be shippable across Europe and easy to use to allow beekeepers to install them inside the hives. And the samples needs to be extremely sensitive, selective and robust to allow detection of atmospheric pesticides present at very small concentration in matrix as complex as beehives air. And so we have selected passive air sampling methods to allow us to sample without power supply. The samplers need to be harmless to the bees and users, but also strong and miniaturized to be installed inside the beehives. So to sum up, we are developing miniaturized passive atmospheric samplers resistant to bees, ready to use, able to accumulate a wide diversity of chemicals, and to detect small pesticide concentration within beehives atmosphere. To do, we have selected two kinds of prototypes that could be adapted to passive air sampling, the stir bar soreptive extraction and the tea bags that are two thin membranes which contain adsorbative sorbent. These tools can be optimized by selecting sorbents showing a better chemical affinity with the compound of interest. And as you can see here from this graph, around 90% of the compounds extracted by solvent extraction are quantifiable below 10. Solvent extraction allows us to split the extract between different fractions to perform multiple analysis and backup of samples. Here we have conducted preliminary trials in the field to evaluate the ability of the bars to sample atmospheric pesticides. The samples were exposed to during 2 weeks in a wine growing area in south of France. The samplers were analyzed and 8 pesticides were detected. These pesticides show diverse chemical properties and display major environmentally pollinator and pollinator health of interest. For POSH-B, we are using the network, the European network, to dispatch the samplers across Europe, and nearly 90 samplers were prepared and shipped. Samplers were installed by beekeepers on 8 sites. We had 4 oilseed rape sites and 4 apple sites from mid-April to mid-August this year. The samplers then were shipped to France and will be analyzed this year. To conclude, POSH-B is on the way to delivering 2 key monitoring products and technologies that are co-developed with stakeholders and research. And we will see all the results in the coming few months, and watch this space. Thank you. And before finishing, I would like to thank Professor Brown for his trust and all POSH-B partners for the work they have done and they are doing now for the project. Thank you.
Thank you, Dalil, also for keeping up to the time. It has been very
speed.
Yeah, no, very nice. It was very clear and very nice. Are there any questions among the attendees? Don't be shy. Just so here we have Sue. Yeah, Sue Collins. Sue, can you please make your question? Sue? Sue wanted to make us a joke. It's okay. Well, then I would like to ask you a question, actually. These 2 tools that you are, these 2 new technologies that you are putting on the market, let's say, for monitoring, how far developed are they? So, I mean, is this kind of in a proof of concept situation, let's say, or level? So it is an idea, you are testing it, but then it still requires much more validation and much more kind of piloting, or you think that they will be ready to be used by field practitioners by the end of POSH-B, for instance, or by when? I don't know.
Yeah, I think they are in a good level of progress. I think they will be available at the end of POSH-B. For example, for the multi-bee typing was already validated for the honeybees and for the bumblebees, and will be also validated for solitary bees and for some novel species also. So I think even for the passive air tool it will be available at the end of POSH-B also.
Actually, that's the question of Sue. Maybe could you please develop a little bit more about the work that you're doing on wild bees?
So we are using the same tool, but we don't have information about the mass fingerprints, for example, of Osmia bicornis, of the other species, the novel species. We don't have the database of the proteins. This part will be developed later, and it's under developing. But for honeybees and bumblebees, we have, I think, it's available. We have all. Our method is already validated on this species.
Okay, thank you. And maybe just actually in link with the previous, so we have a question. So Filomena Brandau, please. I think you can only, I'm not sure if you can speak, but at least you can maybe write it.
Filomena, you may try to speak now. You should have a prompt that allows you to unmute yourself at this moment.
Maybe Andres take
Filomena, if not, try typing your question either on the Q&A or on the chat, please.
Okay. So actually, if those of you who want to, those of you of the attendees who would like to say something, please raise your hand. Then you will need to activate that you can, so you need to be permitted to talk actually. So please raise your hand first, then we will see that you want to say something and we can activate you for speaking, okay? So Sue Collins also want to say something. I think now you can speak, Sue.
Thank you very much. Can you hear me now?
Yes.
Hello, thank you very much for that very interesting presentation and for confirming that you are currently working on bumblebees as well as honeybees. That's very interesting. And I just wondered how you were collecting your samples. Are you using volunteers or how do you access the bee samples that you need from the wild?
It's thank you for the question. It's from lab experiments from some of the partners within POSH-B. They are doing experiments like MLU in Germany and in Switzerland, also in Zurich. They are doing experiments on Osmia, and we have also Osmia from the field from 8 countries from Europe. So, and all these samples or these hemolymph samples from these Osmia bicornis were analyzed here at Biopark in France.
Okay, great. So they collect them in the wild and they take them to the laboratory and dissect. Yeah. Okay. Thank you very much. So you're using professionals or academics rather than volunteers?
Yes, it's academic. It's the persons from the project, and we trained them to collect the hemolymph with our method, and we did the training several times, and even, we are always in contact with them for the collection. And I will say that for the field, it was really difficult to collect hemolymph from the field and the semi-field. It's not easy at all. It's not like the lab conditions.
Yes, it will be really interesting if eventually you extend to hoverflies and butterflies and moths.
Yes.
Well, there is the invitation. So I'm going to cut you here because we need to move on. Those of you who could not make the your comments or questions, please do them either in the Q&A or in the chat, and then Dalil can answer them in writing. Okay, thank you very much. So we are moving on to Dr. Kobi van Doremalen, who works as a senior researcher on the honeybee health at Wageningen University. So she will present the activities that they have been performing within the Be Good project to monitor honeybee health stressing factors. Now, Kobi, we are seeing your speaking speaking mode. And now we are seeing the good one. The floor is yours, Kobi.
Good, thank you. Good morning, Mr. Chair. Thank you for the invitation to speak today. And also good morning, participants. I speak on behalf of the Be Good coordinator, Professor Dirk de Graaf of University of Ghent, and the whole Be Good consortium, which consists of 17 partners in 13 countries. My name is Kobi van Doremalen. I work at Wageningen Research in the Netherlands, and I'm work package leader of work package 1. So the aim of the Be Good project is to pave the way towards healthy and sustainable beekeeping within the European Union by following a collaborative and interdisciplinary approach. And we do that by collecting data from within and around beehives, as well as socioeconomic conditions, to develop and test innovative tools to perform risk assessments according to the health status index developed by EFSA. And we do that in order in the future to guide beekeepers in their decision-making to keep their honeybee hives healthy. So in short, we make bees and beekeepers' lives more easy. So we do that by creating a European network of honeybee colony data to collect. We develop innovative and autonomous hive monitoring technologies and techniques to be the least invasive measurements possible. We use machine learning to identify relationships between health and colony state measurements. And also we have a very interactive multi-actor and multidisciplinary approach. So we use a lot of stakeholders and also end users in the project. And in the end, we will also focus on knowledge transfer targeting EU beekeeper networks. So for the automated bee health monitoring in the Be Good project, we fully rely in a first priority on the BEEP system, which consists of a BEEP app, which you see on the left to log beekeeper actions and experimental measurements, and also on the BEEP base, which you see on the right on this slide, is a scale for colony weight, and it also includes a temperature and a sound sensor to log colony status in an automated way. And we use these systems under all hives in the project, which consists of multiple tiers. But beside that, we also develop new technologies, for example, vibration measurements using an accelerometer, temperature of frames to have a 3D overview of the temperature in the hive, gas emissions, sophisticated bee counters that also can distinguish between the type of bees that fly in and out of the hive. Pesticide and virus detections. We developed an LFD, like the quick test, the fast test that we do also, from the corona testing. And also the lab protocols developed for detection of bee resistance to Varroa. Sorry, yeah. So in the project, we use a two-step approach. The first is the development of these tools, which consist of the BEEP base, these different tools, and also an EU-wide data health portal where we collect all our data. And the second phase is actually to collect data in the field, which we will do over 3 field seasons. The first season we already started in 2020. That consisted of collecting data from honeybees colonies from 8 different partners in Europe that all have like their own apiary and are focused on bee research. In the second season, we invited beekeepers in 5 countries in Europe on a north-south axis, and those beekeepers are closely supervised and guided by partners in the Be Good project. And the third season, which will start the coming winter, is involves volunteer beekeepers from across the EU region, which means that from the first to the third tier or season, we increase in EU coverage, but we also try to decrease in the invasiveness of the measurements that we do. So basically what we are trying to do as a whole is the operationalizing of the EFSA Healthy Bee Toolbox. And that is a document that was produced by EFSA, which consists of an enormous amount of components in and outside the hive that can be tested to give an insight in the health status of a colony. On the right-hand side of this slide, you see a long list, which you actually do not really have to go through and read it, but it shows you the amount of colony attributes and external driver and colony outputs that we are currently measuring in a first and a second tier in this the Be Good project. And we do this because in the end we want to give automated guidance to the beekeepers in the future, but in order to do that we now need to very closely measure all kinds of components in the honeybee colonies. So the primary focus on the Be Good project is actually focusing on healthy colonies, and health is then determined subjectively by the beekeeper. So we monitor in total 384 colonies in these 3 different tiers, so it's 3 different seasons. And basically we assume that if they deviate from health, they're stressed. But also to test our new technologies and new techniques, we also have 20 stressed colonies. We keep them only in the Netherlands, so they're experimentally manipulated. And those, so we have 4 different stressors, 5 colonies each, and those consist of varroa exposure, food restriction with a pollen trap, experimental queen death or like simulated queen death, pesticide exposure, and obviously we also have some control colonies to test them against. So we are currently still in the process of collecting a lot of data, so I cannot really show you results yet, but I did want to show you some results, also to give you an insight on what kind of things we are measuring. So this, I will show you an example of 1 week of data automatically collected, and that was actually from 13 to 19 September, so very recently, 2 weeks ago. And I will show you the automated data for the weather, the hive weight, and the temperature of the nest of the colony. So this data slide shows the data of a healthy control colony. So on the top graph, you see on the, so all y-axis are just the full week from Monday to Sunday. On the top graph, you see the weather of the, so we have at the apiary weather station. So it shows you like temperature, humidity, air pressure, rainfall, wind speed. So it's just to give you an overview of like we check the weather. Throughout the same week, we also measured the weight of the colony, which you see in the center graph. And this was a healthy colony. It weighs about 35 kilograms. Over the time of this week, it decreased a little bit in weight, so it ended up with 34 kilograms of weight. And you see on Thursday, like approximately 9 o'clock, there was a huge dip in weight of about 4 kilos, and that was because someone worked in that colony and had a check and fixed it, put it back together again soon after. In the bottom graph, you see the temperature of the nest, which is approximately 34 degrees. That means that there's brood in the nest, and the sensor is actually also within the brood nest. And also here you see at the same time there's a decrease in weight. There's also a decrease in temperature of the nest. It reduced to 30 degrees Celsius, and they overcompensated a little bit afterwards. To keep up their colony temperature homeostasis. So, but then if we look, for example, to these food restricted colonies, you see the same weather data, it was the same week. In the center graph here, you see the weight of this food restricted colony. So this colony has a pollen trap on the front. And you see actually that this colony was a lot lighter. So it weighs approximately 20 kilos. Most likely because the pollen trap was on for months already before. And also what you see during this week that the colonies actually reduced a lot in weight over this month. So it dropped from 20 to about 17 kilograms or 18 kilograms. And also for the temperature in the nest, the bottom graph, you see that there's a lot more fluctuation in temperature. It decreases also a little bit over time. So this shows you a completely different profile. And it's our task in the Be Good project to in the future to compare these different groups and also to get insight in how this works and how we can interpret this data. So another thing that we do in the Be Good project is also look at the landscape around the beehives, which we do by floral models. For that, we first determine the major resources for honeybees, and then make dynamic maps across Europe, and that will allow us to predict the availability of floral resources around apiaries that we know where they are. Also, we do surveys and a workshop more for the socio-economic component in the project. For example, we ask questions on how beekeepers and stakeholders define sustainable beekeeping. And all these kinds of data, so within, around beehives and also the socio-economic conditions, we combine to perform risk assessments according to the health status index. But for that, we needed a tool to combine this information. And we selected the APES-RAM model to combine all the data on environment, but also stressors. And we use it to model and predict colony health and guide beekeepers for management.
Yeah, sorry, Kobe, can you read? Okay. Very good. Thank you.
Oh, it says a new share, but I'm stop sharing. Sorry.
No, so sorry, I just sorry. So thank you very much. So we only have time for one question, which is from Agnes Rote, and she says, "Thank you, Kobe, for the nice update on the Be Good. Great progress. In previous sessions of the event, there was the question of the potential effect of digital tools on bees. Did you test that? Any views on this?"
No, we didn't test this as a whole, but we do find, for example, that if you have temperature measurements or accelerometers, those kinds of specific tools, we do look at also the impact on bees. For the temperature sensors, we find that it's the bees will show, they will just not use the frames for the temperature sensors if they don't like it. So in that sense, we do take that along. You can never be very sure on the effect of automated measurements. In general, we assume that it's just less invasive and less harmful than opening the colonies all the time, because you also saw in the example that if you open a hive, the temperature of the brood nest, for example, drops. You immediately see the impact of opening a hive. So an automated sensor will allow you not to open it and just look from a distance.
Thank you. So there are other questions for you in the question and answer. So I ask you and Dalil also, because there is also another question for you, to please write the answer. And I'm going to go.
Thank you.
Thank you, Kobe. So now I'm giving the floor to Dr. Joseph van der Steen, chef, who has been working in bee research for over 40 years and is currently an independent consultant and coordinates the Insignia project. So he's going to introduce us to the project that is using honeybees as environmental samplers in collaboration with beekeepers. Chef, the floor is yours.
Yes, Okay, I'm Chef van Steen, and let me first thank the chair for inviting me for this presentation. I'm going to talk to you about the Insignia project, and the Insignia project differs a little bit from the other projects that we hear, because it's a project for a guideline development for having the honeybee colony as a tool, as a biomonitoring tool for the environment. And we did it, and this was also the objective of this project, by making a guideline very comprehensive with all the aspects of how citizen science approach for guideline development and biomonitoring. That implied that we made citizen science protocols. We also worked on meta-barcoding, on modeling, and I'll show you in this presentation, which is only an overview because of the time limitation. We also tested the non-apis bees. In fact, we did test the honeybee colony as a bioenvironmental tool. And again, the study is the honeybee colony is the tool and the environment is the subject. And hopefully it's launched to a pan-European apiculturist citizen science monitoring study in the future. Let me stress one thing. We did this sampling of the bees completely non-invasively. It was important in this study. I move on to the next. After we had done some preliminary studies in 2019, we did a big study in 2020 in 9 countries. You can show here in every country we had 9 apiaries and we had 10 sampling rounds. We collected pollen every 14 days for 1 day, and we also applied the apistrip, and I'll come to that later, apistrip to detect pesticides inside the hive. The apistrip is a non-passive in-hive sampler. We tested the apistrip for 273 compounds. That's not completely, but you have to make a choice. And here comes the results. So let me start with pollen. The pollen were identified with meta-barcoding, and that means that this technique was improved. And additionally, we also worked on completing and making additions to this European ITS pollen meta-barcoding laboratory. Now, here we have the list of the pollen that we did find in the Insignia project, and let me focus here on the right thing. This is the top 10 of pollen that we did find, and is on family level. And you can see here, looking at the top 4, that the Rosaceae, the Brassicaceae, the Asteraceae, and the Fabaceae are absolutely the most important pollen providers throughout Europe. As we can see, that goes from Latvia all to Greece and Ireland. And this is in line. You see the 4 or 5 major families, and this is also in line in the findings of the CSR pollen study we did some years ago. I focus for some details here on what we did find in Belgium and in Denmark on pollen. And here we have this top 4, as you can see here. And we have a green bars for the rest, that means the rest. And in most countries, we did find between 20 and 30 different plant families that were important for bees and from which bees collected pollen, which is rather restricted given the enormous amount of pollen that are available. But looking at Belgium, you can see here, for instance, this Rosaceae. We have a lot here in spring, which it decreases. And Rosaceae is typical, a family with plants that are crops, agricultural crops, and plants in nature like raspberries. And most likely you see here that the orchards in the environment of the test apiaries were flowering, and it decreases. Looking at the yellow bar, the Brassicaceae, it's not that much, it's increasing at the end, which is completely different in Denmark here, which is a big rapeseed field country, and you can see here there's a lot of rapeseed pollen collected. Looking at the green bars, the rest, it's striking that about 70% of the pollen in Belgium were collected from the rest, And in Denmark, it was only 30–40%, which is completely the reverse, which indicates that Belgium is more small-scale agricultural landscape, whereas Denmark, at least that's my first interpretation, is a largely scaled agriculture landscape with less pollen diversity. Moving on to the pesticides, we did, as I said, you test for 274 pesticides, and we did detect 174 pesticides, which is a lot. And this is an impression of the number of pesticides with the chessboard diagrams, in which you see on top of all the bars numbers which shows the frequencies, how many times a pesticide and more or less a compound, and it's the group of this, I'll come to that later, has been detected. What's most important, if you look at these figures, and I will not do it in detail because it's take too much time, but you see there's a lot of blue, and here's the frequency. No color means that it's not detected, and very dark blue means it's detected many times. So you see here that the frequency of detection is about, when it comes to apiaries, from 0 to 7. Here in Italy, here in Denmark, the same. It's in the 9 countries more or less the same. Same here, when you look at the data, this is the time period that you see here. Also, we have this frequency of the up to 7, and which is striking here. You have here this very dark blue spot on sampling day 7, date 7, which means that there is the detection of 0 is frequent. So that means there's many times not detected. And this also reflects here when I look at this bar here in Denmark, which is Denmark, you see the very low number of pesticides in general are found here. What you see here more or less in Latvia and in Denmark is the increase of amounts of the number of pesticides, which has a peak here in the summer, and it's more flat in Denmark. And the median of the number of compounds that we detected is here, ranges from 3, 4 up to 10, and it has also outliers up to 16. And in some countries, we see outlines up to 21. For instance, here, this one has an outline which is about 20. So it is about pesticides, a lot more to say, but just to give you an impression of the frequency of the pesticides. And this brings me to the next one. When you check all the pesticides that you found, you know, we did check more, because not every compound that we checked is a pesticide. We also had biocides and veterinary products and pharoacides, because we did in-hive detection, and you have that. When you look at it, you see about 60 to 70% of the compounds that we did find has an approval status, and about 40 did not. Does that mean that this 40% or 37% is all non-approved application? No, it's not, because we did see that there's some compounds like imidacloprid or fipronil, which are forbidden as a pesticides for application where they can get in touch with bees, are approved biocides. So in case we did detect them, we consider them as an approved biocide. But we also detected wax contamination. For example, propagit was found in many, many countries, and it's absolutely forbidden for every application in the European Union, but it's a pharoacide in Asia. We also did detect DDT and other stuff, which is known that is part of the soil archive. So there we can see that there are many ways pesticides enter the colony via the wax, but also via the soil, which goes by dusting and maybe other ways. We also did see some nanoparticle collection, and water collection is most likely also an important influx of pesticides, especially biocides. You see here that this image, this is an overall image, this of the 9 countries is more or less the same. brings me to the modeling. The modeling, we did result the modeling, we did develop the modeling for pesticide risk exposure and for pollen diversity, and it was done by machine learning in the Wageningen University, based on the residue data and the pollen and the greener land use, cover data and weather reports. We could make a spatial and temporal model for predicting the pesticide exposure and here the pollen. And let me just briefly show it. When we come to the predicted model of this pesticide, you have green is very safe, red is very dangerous, a lot of risk of exposure. And here in May, in Denmark in this case, is orange-green, so there's a risk of exposure which decreases in the time of the year. For pollen, green is best, dark brown is bad. We have here yellow, so in between, that means that's available of about 5 different pollen families, which increases towards June and then decreases towards August. We have this kind of predicting pictures of the 9 countries of the insignia. As I said in the beginning, this project was made to make a guideline, and we did make a guideline. And actually, this guideline was published this morning on the European Pollinator Information Hive. This is a guideline. It's about 19 pages guideline, which has a rationale, which means every in and out of how to use honeybee colonies for biomonitoring are discussed. We have here how you organize a citizen scientist study, because that's a very special way of doing it. And we have very clear protocols and the methods, and we have leaflets and a poster made of it. And that brings me to the end of this presentation within about 10 minutes. And let me thank you for your attention. I'm happy to take your questions.
Thank you, Chef. We have indeed one question. So it's actually a question for clarification. The percentages of that you have presented, they are mainly for Apis mellifera. Yeah, because it was absolutely a honeybee apiculturist citizen scientist study, and we also did some bumblebee tests, but the time was too short. So we're now talking about Apis mellifera. Yeah. Okay. And then it's a second question, and with this we pass on. It's right, Michael Rubinik. If it is the intention to use honeybee colonies as biosampling tools for pesticides in the environment, did you consider that this might negatively affect the marketing of Hive products by particularly commercial beekeeping operations once results are published to a wider public, even if pesticide concentrations might be far below maximum residue levels? Let me be very clear. This study was made for detecting what pesticides are in the environment and enter the honeybee colony. Of course, we did some studies, and we hope to do that in the next study. There is a significant difference in what you see in these apis strips that we used to detect pesticides in bees and in honey. And the amount in honeys are very low, but that many monitoring studies did show that. But that's absolutely a point we have our attention for. Yeah. Thank you, Chef. So you have another question in the question and answer. So can you please write it? And then I'm going to move on to the presentation of Dr. Rainer Oppermann. Rainer Oppermann, thank you for switching on your camera. Is senior ecologist and head of the Institute for Agroecology and Biodiversity, IFAB, in Mannheim, Germany. And he is going to present us the work on landscape evaluation that they are doing within the EMBAL project. Yeah, Rainer, the floor is yours. Can you share your screen?
Yes, thank you for the introduction, and good morning, Mr. Chair and all participants. I'll present you, give you a presentation on the project EMBAL. EMBAL means European Monitoring of Biodiversity in Agricultural Landscapes. That's a project which we are working on together with partners, EFTAs and the Austrian Umweltbundesamt, Environmental Agency Austria. And that's a project which is deals with monitoring of agricultural biodiversity, though not especially with pollination, but pollination is also an important outcome of this project. So the purpose of this ample basically a field survey, and it delivers a robust tool for monitoring the farmland biodiversity and nature value across European member states, and it aims to provide data on land use, biodiversity, and ecological value of agricultural landscapes, and is comparable between regions and over time. So we are actually we are in a pilot project, and it's planned from the side of the DG Environment to do a rollout in next years. Only areas under agricultural use are surveyed in detail, so arable land, permanent grassland, permanent pasture, permanent crops, and landscape elements. And non-agricultural elements are not surveyed in detail. We have a grid sampling design, so there's a grid of 2 kilometers across EU 27, and several plots are selected, and the plots are each 500 to 500 meter, so 25 hectare in size, and the minimum coverage of agricultural land is 10%. And on these parcels, on these plots, we do a mapping on the one hand side. So all the 25 hectares are mapped, land cover, types of land cover, landscape elements, patch density, and so on, and nature values. I come back to that point later. Then we do on specific points, up to 5 points in such a plot, we do transects with specific measurements on species richness, species composition, and structure of vegetation, also pollination potential, coverage of wild plants, and we do a photo documentation to allow a cross-check with data of the surveyors and documenting changes over time. So it's a threefold approach, this EMBL methodology. I show you this. This is one example of such a plot, and we have numbered all the parcels, and for these parcels we survey, we record different things, so land cover, different types of land cover, irrigation, flower colors, so as a proxy for the diversity of flowering plants, the width of strips, the flower density, and also in arable land, the crop coverage and the wild plant coverage, and in grassland, the type of vigor and the graminate forb ratio. So that's for the parcels. And we do further some transects. I told you up to 5 points in such a plot, and we do always a border transect and a transect inside the field. So that applies to arable land and to grassland. We monitor certain key species or key species groups. For example, in grassland, we have red flowering trifolium species, so clover, or vicia species, or medicago, or apiaceae, and so on. So there are 12 plant species groups. For arable land that are up to now that are 40 species or species groups, but that will be elaborated later on if the first rollout comes in order to reduce this number of species. Flower density is recorded with such a scale from 1 to 5, so none or hardly any flowers to a dense flower carpet. Also, the landscape elements are monitored with nature values between 1 and 5. 1 is, for example, an asphalt track in the landscape, as a landscape element, and 5 is a species-rich and structured landscape element. Here you will see, for example, these different types of strips, grass strips alongside of field passes, field tracks. So we have different elements which stand for the pollination potential, especially flower density, number of flowering colors, and the landscape element, nature value, which is we have a scale of from 1 to 5. On transect level, additional kind and number of indicator species and the cover of legumes. It's a specific tool in addition. In some, we have different parameters to record the pollination potential and the nature value. So referring to the species richness, to the coverage of flowering plants, and to the nature value of landscape elements, and that can be synthesized, can be aggregated to plot values. So here you see in this case the different values of nature values of different parcels in a plot, and they are all aggregated to a plot value. In this example here for this plot of 500 to 500 meters, this is a value of 7.2 and on a scale from 1 to 9. So it's rather extensive land use with a lot of different plants and of a lot of different landscape elements which have a high value, and therefore this is a high value here, 7.2, and only a few fields like this here has a very low value. This is an arable field, probably with intensive use. So this is a proxy also for the pollination potential with different values, but not measured in detail the specific value for different pollinators, but in the whole it gives an overview on the nature value across the European Union. And as I said in the beginning, it is planned to do a rollout in the next few years of this methodology, and that will deliver and provide data on the pollination potential in European agricultural landscapes. So thank you very much for your attention, and I'm open for any questions from your side.
Thank you very much, Rainer. It's very nice, and yeah, you also did stick to the time, so thank you very much. Are there any questions from the audience? Yeah, we have one hand up. Andres, so it's
Filomena, I'm not sure if it's from before or if you still have a question, but if you still have a question, you may speak now. You may unmute yourself and speak. If not, Janko has also a question, so we'll give the floor to Janko. So Janko, you can unmute yourself.
I'm sorry.
Put on your micro.
Janko, we could hear you for a while.
Yeah, I'm sorry. I just keep put my hands on. I really like this presentation. So maybe also my coworker is attending here, but we have some similar project called Bee Diversity. So we're trying to arrange. Maybe your talk will help us to do better job. But just mistakenly put raise hand. So since you noticed that, then I thought you. Thank you. It was great.
Thank you. Filomena?
Not Sue Collins also has a question.
Yeah. Then Sue.
Hi, thank you, Rainer, very much for that really interesting presentation, and we look forward to the rollout of the MBAL network, because linking butterfly data and other pollinator field data with land use and management data is a really important issue for assessing conservation effectiveness. I just have one question about your inclusion of flower strips in landscape elements. We heard yesterday that it's really important, the management of the flower strips, and if they're short-lived or if they suffer from pesticide drift or they're just very, they're ploughed out really quickly, then they're not actually as valuable as they could be. What you need is long-lived, long period of flowering. So I don't know if you're able to take that nuance into account in your system.
No, we don't take this into account. We only monitor at one date during the main vegetation season, and of course, from that we don't know how the flower strip will be managed over the year and in the autumn. So it's only a rough methodology, and that is left to management. Restrictions or recommendations in the member states. So we can't do that with this rapid approach because it's only at one time during the year.
Thank you.
Filomena?
Okay.
I mean, well, if you cannot, it seems that you're having a lot of trouble. Maybe now? Can you unmute yourself? Yeah, but it seems it's not really working. So please write your questions into the chat or into the question and answers. Sue, you still have a question? Okay, thank you. Great. I just would like to ask you, what are the next steps for the MBAL project?
Next step is the rollout. The European Commission has announced the rollout for this year to come. So we are curious if in the next few weeks, possibly, there will be a call for tenders to do the rollout in the European countries across EU 27. So we hope that the first MBAL rollout can start in 2022, and then it will possibly provide data across all the member states. Of course, it's planned to first only a small pilot or with relatively few points, but in all member states. So in order to provide data for each member state, well, that's the plan, but we are waiting for this call for tenders now. But the plan is to already provide data before the new agricultural policy will start from 2023. So therefore, it would be very important that this MBAL starts in 2022 to have already data from 2022, and then to compare the data from 2022 with data later on in order to show the effects of agricultural policy, hopefully in the positive sense that the new agricultural policy will improve the ecological basis of the agricultural landscapes.
Mm-hmm. No, definitely, I think you have a very important tool there and promising tool to verify the efficiency of all these future measures. So thank you very much, Rainer. I'm going to give the floor now to Dr. David Roy, who is an expert in environmental monitoring and assessment. He led the European project ABLE, Assessing Butterflies in Europe, and is co-leading now the Spring project, so strengthening pollinator recovery through indicators and monitoring. Yeah, Dr. Roy, the floor is yours. Thank you.
Many thanks. Good morning, Mr. Chair, and welcome all participants. And I'm very grateful to have this opportunity to present the Spring project. I'm coordinating this with Joseph Satellet at UFZ, who's the overall lead of the project, but I'm presenting today on behalf of all the partners. So in terms of why it's important to monitor wild pollinators, I hope to this audience I don't need to convince you, but it's really central to our understanding of the causes of change and measuring progress against policy objectives and understanding how we manage the landscape and use resources. So a crucial element of our ability to understand change, but also monitoring has a much wider potential, much wider benefits, particularly when it involves community science, citizen science, with a role in educating, involving and engaging the public in the issues of pollination, pollinators, but also wider biodiversity. And there's some nice evidence that monitoring clearly comes at a cost, but it has clear economic benefits when measured against the return on investments in terms of, particularly for pollinating insects, their role in pollinating our crops, but also supporting wider research and society's aims to understand the environment. So it can be very cost effective at delivering in multiple ways.
Sorry, David,
yes.
Are you using your presentation?
Yes.
Yeah, because it's not sharing.
Ah, sorry. Let me share my screen. So
now, yes.
Let me go back to the beginning so you can just see the.
So here we are seeing the speaking.
Is that okay?
There you go.
Yeah.
Yeah.
So I was on this slide where, so yes, there's clear benefits to monitoring wild pollinators. And the final point I was going to make on this slide is that with growing awareness of insect declines, monitoring is a vital part of the solution to understand where change is happening, the magnitude, timing of change, and understanding why it's happening to identify solutions. So with that in mind, the EU Pollinator Initiative, one of the actions was to establish an expert group to define the approach to monitoring pollinators in Europe. So this group was expertly led by Simon Potts, with great support from the JRC through Joachim Maas and Chiara Puls, and involved a range of experts across the whole of the EU, both directly within the experts involved in the expert group, but we consulted with experts across all member states. So a collective community view on the best approach to monitor pollinating insects. And the very detailed work, very comprehensive report on this expert group was published in early this year, the beginning of this year, and is available via the available online. Over 300 pages, giving lots of detail on the rationale for the decisions that were made and the approaches that are being recommended. And I'm just going to go through some of those and how the Spring project is implementing or piloting these recommendations. So the scheme overall or the recommendations for monitoring wild pollinators in Europe composes a core scheme, which is a set of recommended methods, both the minimum recommended methods and complementary approaches, and then a range of additional modules to cover other aspects of pollinating insects in Europe. So the primary field methods that were recommended or are being recommended and being piloted by the Spring project are transect-based approaches to target bumblebees and other bees, butterflies and hoverflies, and to link into the established butterfly monitoring schemes. And you'll hear about this afternoon from a talk about the ABLE project. And this is standardized transects involving both professionals and volunteers. There's a strong citizen science role in butterfly transects and can be applied to other pollinating insects. And this is going to deliver us species abundance counts in a structured way. And to complement this is pan traps. These are water traps that attract insects and typically give us good estimates of the diversity and occupancy of wild bees and hoverflies. And we'll be standardizing how these traps are deployed across the EU. Typically involve professional field sampling, but can involve volunteers in setting and collecting the traps. But the identification of these insects from the traps requires a level of expertise. And then within the Spring project, we're also piloting complementary approaches, which is seen as being important additions to the core parts of the schemes, particularly methods to assess moth populations. There's increasing evidence that these are really important pollinators to complement bees, butterflies, and hoverflies. So night flying insects can provide a complementary role. And we're providing a standardized approach for volunteers to get involved in running these traps across the EU to provide really valuable information on the abundance of these insect groups. And the final element of sampling that we'll be piloting in the spring project is malaise traps, which again operate across various member states in Europe and has been very important in understanding trends in insect populations, give a wider assessment of insect diversity, and can provide estimates of the biomass, the total numbers of individuals within this trapping regime. So we'll be looking to standardize these approaches and trial them across several member states. So the spring pilots, in terms of what we're focusing on, in the dark highlighting are the main methods of the recommending pollinator monitoring scheme that we'll be piloting. We're also looking potentially at other flower visitation methods that can be used for wider citizen science engagement activities. So that's the recommendation of the expert group. So the spring project is just started. It's a parliamentary preparatory action. We'll be mainly undertaking fieldwork in 2022 and 2023, covering 200 pilot sites across all member states. And this diagram of our work patches, I've mainly talked about the field sampling elements of the minimum viable scheme and the additional modules, but we will be investing a lot of effort and time into building the taxonomic capacity. Clearly, to deliver this sampling is going to need lots of expertise, lots of involvement from citizen scientists, from expert taxonomists, and we're providing materials and training to help build that capacity across the EU. And we've also got a series of activity on citizen science approaches, building on the work on butterfly monitoring, but also looking at other approaches for pollinators. And a crucial element for us in being able to deliver this project involves 19 formal partners, but really would value and would need contributions from a range of other organizations, particularly member states. So we'd welcome engagement and contact with member states who are already running schemes or planning schemes or would like to support these pilots within the spring project. So we really appreciate your support. And then the final slide is just to put spring in the wider context of this, the EU Pollinator Initiative, from going from a design phase, the expert group that I've already mentioned, and a whole series of projects that are now running or just starting to pilot and refine the methods and build capacity to then roll out the scheme more widely across the EU. So spring sits alongside particularly the Orbit project and the TaxoFly project, which are building up taxonomic resources, and we're very closely collaborating with those projects, and also the Red List assessments, and then a range of Horizon Europe funded projects and other initiatives that we will be working alongside. So thank you very much.
Thank you, David. So we have one question by Adam Reichhold. So are the sampling procedures limited to the insects, or do they have insect-plant interaction-oriented aspects as well?
So within the minimum viable scheme of pan trapping and transects, they're not predominantly looking at insect-flower interactions, but we're still discussing the role of transects in collecting that data. But we are potentially looking at citizen science approaches, flower counts to provide additional interaction data.
Thank you. I have a question actually. So since this is a preparatory action, if I understood well, so the idea is that eventually member states will interiorize, let's say, so take it on board. So have you, so do you have the members, let's say, member states, let's say, already there to intake, let's say, all these procedures and all these,
I think that's a crucial point, that it is preparatory action to provide the recommendations and the approaches for member states to then apply the scheme in future. So already within the expert group process, we've had good engagement with member states, and we would look to continue that within this current spring project to engage with member states to understand their capacity and how we can support them to take on the scheme in future. And we particularly work with the Commission to have those discussions and think about the future plans for the full monitoring scheme for pollinating insects across the EU.
Thank you. Are there any questions from the other panelists or the attendees? No. Okay. So with this, actually, I would like to open the floor to discussion. So we are going towards the end of the meeting. So now there is some time to discuss openly among with the audience, with the chair and the panelists. So I would like to invite our chair if he wants to, make any questions, suggestions, launch a discussion. Martin?
Yes, I'm here. Let me just turn it on.
Sorry, Martin, just can the panelists please switch on their videos?
So no, for me, that is the one important question to all the panelists. And where can we help you? What are the concrete actions that you would like to see also from the European Parliament? The needs that are, so to say, covered or they're also not covered by the projects we have with us, and where do you see us kind of helping to facilitate? So any recommendation towards the European Parliament would be highly welcome.
So I think from the spring project perspective, it follows on from the question that Noah just asked about involving the member states in working with the spring project to understand what's needed to monitor wild pollinators in the long term, what capacity and what support is needed to help the member states and take on this monitoring scheme in the long term. So ideas for,
sorry, just to follow up on this, is it still possible for member states to kind of hook themselves onto the spring project? Because this is something I was struggling that the Slovak authorities didn't really join, but then kind of I hear from the state secretary on Monday that, yeah, we'll do pollinators monitor. It's like, that is an EU-wide project. I pushed to get it through and get funding for it. And, you guys, when I was inviting you to join, didn't really react. So is there a possibility, not for the Slovaks only, but for anyone else still to kind of get on board?
Yeah, we're already in touch with some schemes that are starting already. You mentioned the island scheme, and which is very well developed already, and schemes starting in Sweden and Spain, for example. So I think the key thing is to try and standardize the approaches. So those new schemes, I believe, are adopting the recommendations from the expert group report in terms of methods and approaches. So we're starting to engage with those member states that already. So I think it's welcome that they're starting already, and we can work with them to provide additional support and advise on approaches and methods.
Can I ask just one thing? So there is now in the afternoon, we are going to have a session about citizens initiative also for monitoring. And there are many like NGOs, natural NGOs also developing these in different countries. So I guess they are part of your, these kind of grassroots initiatives, they are part of your network, maybe, I don't know.
Yeah, we're building on particularly the butterfly citizen science approach for established methods for transects across the EU that are delivering indicators at the EU level already. So we will be further developing those methods within the spring project. And we've also got a piece of work to to evaluate how citizen science can support wider pollinator monitoring of wild pollinators. So we very much welcome the interactions with the ongoing projects on citizen science. So I'll be really interested to see the talks this afternoon.
I would like just to add, there is a contribution from GRC, Chiara Polce, that says that, well, thank you, David. I can confirm that the GRC will continue supporting the implementation of the monitoring scheme in consultation with experts. So these are good news also from the GRC. And that's very good. I don't know, Martin, you want to say?
I have some concluding remarks, but I still have a few more questions. I just don't want to be, the person that kind of takes all the space in the discussion. But if I may just kind of randomly, for me, the question is concrete question to Rainer is if do you as a member, as a consortium of MBAL, coordinate with other DGs? Because I think this is something which it's crucial to see, is especially when we look at CAP, and therefore that kind of the DGs really work closely and don't end up in silos and administrative fights between each other.
Excuse me, I didn't get exactly the question.
The question was about the cooperation of the MBAL consortium, or coordination with other DGs.
Yes, we have a project group in this project, and so the other DGs were involved in the process of developing MBAL. So especially DG Agri and DG Climate, and I think these are essential, and the GRC in ISPRA were involved in the development of MBAL project.
I have questions from the audience. So Agnes Orte is asking if there is any initiative among all of you to promote interaction, synergies, and collaborations. This is a question to all of you. Jacobi.
Yeah, so I think from the Be Good perspective, there is an opportunity, for example, to collaborate with the project of Insignia, the monitoring, because I think these monitoring techniques could well be also integrated in systems like the BEEP system and the Be Good system to give a good to get a good insight on the status of the landscape to which the bees are exposed, but also maybe inside the colonies. So I think there's opportunities to collaborate for such purpose.
Anyone else wants to add anything? Okay, yeah, Katerina.
Maybe I can add that for us at the EEA, the dream, if I may say it, was to have monitoring and an indicator on wider pollinators, starting, of course, with the butterflies. So actually, for us, the Spring Project is a ray of hope. We are following it, and we very much hope that the involvement of member states and all different organizations, NGOs, etc., will be big, because the point that has been raised today by several people is that this the monitoring needs to be streamlined, because especially when the trends are declining, when the trends are negative, the results are very often questioned. So from the point of view of our dreams earlier, I think it's a good moment in time where indeed different groups and also member states can join the Spring Project, but there are also very interesting Horizon 2020 projects, safeguards, and so on and so forth. So I would like to add this more optimistic note also from our side. Thank you.
And from the audience, we have Sue Collins, who wants to make a statement that is very right.
Thank you. No, Martin, you asked what else the European Parliament could do to help. And one of the things is to encourage or call strongly for member states to make their IACS data, that's the land use and management data held by the agricultural authorities, open to the public for researchers to use, because we are trying to assess conservation effectiveness. We know the outcomes, we know the butterfly data, and we'll soon know the pollinator data for different locations. And what would be good, as well as the MBAL results, which are important, but there's a wealth of data collected with public money by the agricultural authorities, and lots of it remains confidential, and it would be very good for that to be open access so that researchers and others can do proper conservation effectiveness studies.
Thank you. So it's a very valid point. Actually, I think we are shockingly non-transparent on a number of issues in Europe when it comes to the impact on the environment. In Slovakia, we have the access to information law that says what is not secret, and there is official list of trades of state secrets, and you can check, and or trade secret must be public, and information about the environment cannot be declared as a trade secret. And I think this is something which I would like to see Europe widely used across the Europe, because indeed this is about all of us, and this is something where I don't really see the need for confidentiality. So thank you for the input.
Rainer, you want to add on that?
Yes, and another point would be that it is possible for the monitoring to get access to the land, and in some or several member states it's very difficult to get access to the land. So that depends on the landowners if they allow for you to monitor. So and for example, in intensive farmland you may not enter, and so that will lead to wrong results. And that could be an initiative of the European Parliament, and that for these European purposes of monitoring, land access should be possible across all member states. So if the European community pays for agricultural subsidies, it must be possible to do other monitoring across Europe, and may not be forbidden by landowners to enter the land just for monitoring once or every 3 years or so. And so that is also an important point regarding the monitoring. But on the other hand side, regarding monitoring, that's only data, and up to now, the agriculture policy doesn't follow the data of scientific evidence that there is a need for more protection of nature. So that's still a point which we see. We provided lots of data, and they are important, and I also work on this data, but the process from the knowledge and from the scientific evidence to acting for nature conservation is a long way.
Thank you, and it's a very valid point, and it's something which would be, we will keep that in mind because of the pollinator indicator that we managed to push into the CAP pending approval of the trialogue outcome, both in the council and in the parliament. It's going to be up to the member states to kind of go and design in detail and provide technical assistance, and it could be something to also, because this week I saw that the Commission was stating that they really want to properly check the strategic plans. So this is something which, if we have a pollinator indicator, then obviously to be able to assess whether it's being done needs to be monitored. To monitor, one needs access. So, it sounds logical, but I get your point that the logical things are not always happening. And second, listening to the science, honestly, this is something that I find sometimes a bit frustrating when you hear, about the excuse of, oh, you know, it's the activists who are kind of pushing the European Union to come up with these crazy things. Actually, it's science. It's science that's been telling us for years we have a problem, and the politicians didn't listen and still don't listen sufficiently. And it needed the pressure of the street to basically get us listen. And I think this is something which is to our own shame, and we have to be reminded of it, because the science the scientists, in my experience, you are always very cautious and careful in the communication of your outcomes. And then what we see is that actually it usually turns out to be the worst case scenarios, because it's very, very cautious. So hopefully we can prevent the worst case scenarios in the case of pollinators. Otherwise, we will have a big problem.
Martin, I'm taking one last comment from the audience, and I think afterwards we can go to the closing remarks. So Jürgen Binden from Germany, he says that we need to encourage the Greens to take the agriculture ministry. Please write and call Robert Habeck in Analema, Barbuk. Oh. If voices from abroad arrive here. Okay. No, I think it was just a recommendation for strengthening and encouraging the activities that we are saying. So I think we can proceed to close the event, unless
concluding remarks.
Yeah, unless Sue has anything to say, because she still has her hand up. So I don't know if
I have something to say to that. A request to Sue, if we can coordinate, because I think this is something which butterflies are always seen as kind of the beautiful, romantic creatures, and they are so also for me, but their importance as pollinators is so vastly understated, and they've been so much missing from the nature. You know, I remember when I was young, which is becoming a long time ago, but not that long, I'm young, that, you know, walking on a meadow in summer was full of butterflies, and it's not the case really anymore. So I hope that we can stay in touch, because this is something which is important. And yeah, so you have here a question, sorry.
Thank you. Can you hear me? Yeah. There are two other points that I wanted to make. Katharina showed the very important Article 17 data on pollinator habitats. We've also analysed the grassland butterflies, which shows exactly the same picture. They're in a dire state, even though they're of European importance. And so I would like to ask the European Parliament for strong support for setting a pollinator recovery target in the new EU binding legislation on restoration that is promised from the EU biodiversity strategy. So it's really important to have that commitment, because then member states will invest in the necessary measures. And we know what those measures are largely. We know what will work. And the second thing is the point you make about, the point that Rainer makes about politicians and policymakers not taking up the science is absolutely my experience of 25 years of working with the European Union and other governments on science policy interface issues. And one of the things is there's a vast array of scientific research available to experts through ResearchGate and elsewhere. I can't understand and absorb it all myself. And farmers have got no chance of knowing what the latest research shows for their patch of land. And I think the European Union could do something really valuable in bringing together all the knowledge that does exist in the scientific community and the published grey and peer-reviewed literature to show and put it in a usable form so that farmers and land managers can actually access the best science on what might be effective for their land. So I think there's a real, and then the dialogue between scientists, policymakers and social scientists to change the mindset so that we get transformative change. That came out yesterday in the workshop on agriculture. We need a mindset change to achieve the IPBES, calls for people to recognize there's a crisis. We don't have that much time. So I think the European Parliament can play a really important role in stimulating political will to act with urgency and scale.
Thank you, Sue, for these needed words. They were very good. And with this, I think, Martin, you can do your concluding remarks.
Thank you, Sue. We will try. I will try. I cannot speak for all 700 of us, but given the support for the decisions that we're making related to pollinators, I think this is something which is having a good response from my distinguished colleagues. Now, let me also really thank you for this session. And I'm sure I'm going to miss some of the things and some of the comments, but I'll try to capture at least some of the essences. And first of all, let me really thank EA for the presentations in general, within the ENVI committee, but also here, and for your work, Katarzyna, and the work of your colleagues. It's always kind of frightening almost to hear the data, but I think this is something what we really must hear, and we need to keep on hearing. Dalal, the contribution and also the reactions really made it clear that we are getting quite a number of scientific data and results, but yes, we need to be better in cooperation among us and among your experts. I think this is really in regard to protection of broader spectrum pollinators. This is really essential. Now, Kobe and Joseph, 174 pesticides. It's quite a number, and a bit scary number. And I'm really curious on what numbers are we going to see when the Commission rolls out the follow-up of this project, which they should have, and hopefully broader sample range and geographical coverage across Europe. One of the crazy thing about the hazardous chemicals and pesticides in particular is that the testing, given that it's actually a targeted testing, doesn't reveal so many of them because we don't have often the broad spectrum. We don't look for everything. When you look for a certain chemicals, then does it mean that the others are not there? It does not. You just don't look for them. And this is something we really need to get better at screening. Now, thank you for the information about the monitoring of the biodiversity in the rural areas, and I'm looking forward to the rollout by the Commission next year. I think this is something that hopefully will also give us a better picture. David, thank you for the presentation of the Spring Consortium. And I'm happy to see that it's large, and I'm really going to be following it closely and looking forward to see the outcomes. And last but not least, one specific request from my end. Although I know that we are talking about pollinators this week, and I believe that this is something which is crucial to pollinators, and that's soil. One of the initiatives I'm really pushing for is to finally have not only the soil restoration, but to have a legislative framework for soils. So just like we have it for air and water, I think we need this common framework for soils, because it's also important for the protection of pollinators. Not all the pollinators live in beehives. Lots of them, have their homes in the soil, and they are being affected there by, for example, the pesticides, but all the other things that we do with and to the soil. And this is something where it would be really great to, and this is for the Commission. I hope that you're listening. That the Parliament has called for a legal framework for soils, not just the restoration, the overall legal framework for soils. And we will keep on pushing until you will present it. And I'm really hoping to also get a broader support and continued support, because there's been in the past, from the science community, because I honestly believe that we need it. And as the data from the EEA are clearly indicating, it's about the time. And I know that we didn't manage to get it through past, but I believe that now we have a situation where we indeed will get it through. And Kobi and Dal, I forgot to thank you too. So also thank to you two for very insightful presentations, and I'm looking forward to future collaboration.
Thank you, Martin, for these concluding remarks. Yeah, and I think, I mean, there is a lot of collaboration that will, if we collaborate with everything, like this is going to be a massive breakthrough, because it's a lot of things going on with lots of data produced all over Europe. So I would really thank you for chairing that well today's session, and also your office for the super nice collaboration and constructive collaboration for the organization of the week in general. I would like to thank the speakers for being disciplined and keep up to the, I mean, send all their presentations in time and everything. It has been very nice. And I would like to thank the attendees for their active conversation and sending off comments and questions. With this, I would like just to finish with some information. So all the presentations and the video, it will be available on the website of the Pollinator Week after after the session, let's say, when we will make all the resources available. So if any of you want to come back to the video or to the presentations, everything is going to be there. And with this, I really wish you a bon appetit. Allez, take care. Bye-bye.
Thank you very much. Bye-bye.
Oh, guys.